So it has already started. Maybe there is a time lag, when we start here and when it appears in the YouTube channel. Okay, so I'm sure all the students are logged into YouTube and to Cisco WebEx. And thank you very much, students, teachers, and parents for coming and making this a huge and successful event. And I will just like to share a short glimpse with you. I just want to show you, how and what we do when you happen to come to NCL. And last 2.5 years, unfortunately, we were not able to get you here. But I'm sure now you will start.
Yes, ma'am. Yes, ma'am. Ma'am. Yes, ma'am. Yes.
Slow, very slow.
Okay, hello. So I heard there was no audio for the video that I played, but there is a background music on it, and you can please go and visit our CSR Jigyasa YouTube channel and watch the video. You will be able to find yourself and you will enjoy that. It's a small short video which we made. We are also having a very active virtual lab which we have planned and also launched last year in November 2022, 2021 November. And you can go through our Jigyasa, virtual lab, and you can go through the activities, you can participate. We recently had a very good program on Jigyasa Vigyan Mahotsav. That was a science competition. And we had about 30 to 40,000 students who registered, and they participated, and 75 top winners were also announced recently at the hands of our SNT minister, Dr. Jitendra Singh ji. He had given away awards, and we request you to kindly come and attend and keep playing and keep using that website of ours. you will also find soon, maybe in a month or two, you will find that the awards which students have won, their activities are also placed on it. And very soon we are also going to launch what you can say, a sign language for the students who are deaf. So we are going to have that initiative also, and it will be enabled on our website. Work is going on. In fact, the videos are also ready. About 20-25 videos are ready, and we will be putting them also on our Jigyasa website platform. Okay, so with this Jigyasa information to you, I would like to now introduce the speaker of our day, and he is very much here, Dr. Chiranjit Chaudhary. We are very lucky and we are very honored to have him join NCL very recently, and he's a senior scientist and he belongs to the Biochemical Sciences Division at NCL. His areas of expertise are biotechnology, microbiology, proteomics, structural biology, synthetic biology. And he has obtained his PhD in biotechnology from Indian Institute of Technology, Kharagpur in 2009. In 2010, he had joined a sister concern of CSIR, that is Cellular and Molecular Biology at Hyderabad, CCMB, where he was a DST, that is Department of Science and Technology Research Associate. And his study at CCMB Hyderabad was focused on characterizing outer membrane vesicles of a plant pathogen. He has got a lot of exposure and a lot of research going on with these kinds of bacterias and similar kind of work that he has done, and his publications, he has got a lot of publications in repute journals. In 2011, he has moved to University of Notre Dame, Indiana, USA as a postdoc research associate, and he was engaged in understanding the de novo recycling of bacterial cell wall biosynthesis. From 2012 to 2017, he served as a postdoctoral research fellow associate at Iowa State University, Iowa, USA. His study at Iowa State was to dissect the fundamentals of microcompartments assembly, encapsulation, and permeability property and engineer them for prospective synthetic biology applications. His research was greatly acclaimed in several international journals, like Analytical Genetics meeting at Alta UT in 2013, Protein Society meeting held at San Diego in 2014, and many more such accomplishments are there with him. You can, just Google him out. You will find his complete details of his profile on our CSI National Chemical Laboratory website on the academic side. And he has returned to India. You can see, carrying the prestigious Ramanujan Fellowship Award, and he joined the MIT Indian Institute of Molecular Medicine and Stem Cell Research as an associate professor in the past. So now we welcome him, and we are very happy, Chiranjit, that you have joined us, and thank you for taking your valuable time and coming and reaching out to the students of Jigyasa. So with this short introduction, I would like to now hand over the platform to Chiranjit. So Chiranjit,
thank you. It is a very nice introduction for me. I am overwhelmed to see a lot of participants are across the Pune city, maybe the outside of the Pune city still joined over here. So Wafia, can you please, give me the access to share the screen?
Yes, I will be doing that. Yeah.
Okay. Okay, so today I'm gonna present one just basic concept of the food and microbe, how the microbes basically influence in the food production as well as food poisoning diseases. So that is for the school student. So generally concept of the health and hygiene, that's my topic here. I'm gonna share the screen. Is my screen visible?
Yes.
Okay.
Presentation mode, Chiranjit. Yeah.
Yeah,
Perfect.
I made a presentation mode. So, good morning, students, principals, and headmasters, and all the dignitaries who joined today for this lecture. Here, the lecture is microbes and food. And what are the food? I mean, what do you mean by food? Food are any substance that actually gives you nutritional support to the any kind of organism, not only to the human, from plant to everywhere. from cattle, any animals are getting the nutritional support like protein, carbohydrate, fat, vitamins, and that will serve those for the growth of certain organisms. So that is called a food. And the microbes also have played a crucial role in producing certain food items. So however, the microbes sometimes serve as a like foe. Those are enemy for any kind of diseases, right? But without the microbe, can we build up a food? So there is always like just debate like microbes are friend or foe. So food microbes are always on the menu, so that people are seeing over here. You can see the slide. All the just a minute the how the foods are looks so what is called stale. It is not at all useful for the consumption. However, without microbe, can you make any kind of cheese, wine, chocolate? No. So without microbe, you cannot have yogurt also in the card what you consume, because the bacteria what generally ferment the dairy product that requires to form having you yogurt. So without a yogurt, without a microbe, you cannot have yogurt, even cheese. You cannot have the bread. So here the bread requires that tiny unicellular fungus, which is the yeast. So without that, it is not possible you can to make the bread. And also other dairy products like cheese, as I said, yogurt, bacteria is utmost requirement. And there are also certain group of people consume some fish items that require the fermentation, that requires the bacteria has to be there, right? Some also use the mold. You can see the mold everywhere in the bathroom. So those are so nasty. But however, without this, you cannot have this kind of fermented food items. You cannot have the vegetables, pickles without the bacterial fermentation. So that you all consume. And it's a favorite item for all the chocolate. The chocolate, the candy cannot be produced without the fermentation of the yeast and bacteria in combination, right? That requires the cocoa bean to be fermented, and it will produce the chocolate item that is all time favorite for everyone. However, there are several reports that microbes actually causing so many disease. Okay, so these diseases are very common in India, and not only that India, even globally. As you can see that there are food poisoning. Many people, many kids died in the recent past here, and most of the cases, this kind of food poisoning thing occurred when there are the bulk amount of the food is being prepared. So here you can see from last 10 years, from 2008 to 2011, approximately that 1600 outbreak reported so far, right? And almost 250 around due to the food poisoning and acute diarrheal disease. Some people also died because of this food poisoning. And where the food is being cooked in a bulk, like in the temple, like in the masjid, mosque, anywhere, the bulk amount of food, as well as in the hostel, wedding venues, everywhere the food preparation that where the lack of health and hygienes are avoided, right? So in that time you will see the food becomes contaminated with some kind of either bacteria or fungus or the spore, and that will cause the disease. So here the food microbiology says in a fermentation that fermented food has been, consumed, before that Christian era, like 7000 BCE, the evidence of the fermentation of the Babylonian the culture, they use the fermented food earlier. Even 3000 BCE, you can see they can manufacture cheese butter by and Egyptian group of people. And 1000 BC, preservation of the stream in the snow by Romans, and they found that's by snow or the by smoking technique, you can preserve the fermented food. At 1600, that Francisco Redi demonstrated that maggot in the larval stage flies uh, in the experiment that to disprove the spontaneous generation theory. So earlier it was thought that the microbes basically generate spontaneously, right, in the food. Where that food is kept over there, microbes, any microbe, any organism generate, but that disproved by the this Dr. Reddy in 1600. In the year 1665, he first time demonstrate that uh, without any kind of component, any microbe cannot be generated. But you work in the larva stage of fly, so that requires the eggs should be there, otherwise larva cannot form, right? In 1683, Antony van Leeuwenhoek, he is the pioneer of the microbiology. He discovered that bacteria and also discovered of course, the microscope is the greatest invention. And 1857, Louis Pasteur demonstrated the microbes actually causing the showering of milk, and he is famous for the novel invention which is called pasteurization. And in the late 1800s, that legislation to protect food quality starts to be enacted by the several governments. And early 90s, that cost postulates came, and relation between microbes and disease. Earlier, that there are several kind of food poisoning things happen earlier, but whether the microbes are responsible for food poisoning causing any kind of disease, foodborne disease, was not known. And then 1920, so there are several outbreak of the botulism that by caused by Clostridium botulinus, that prompts the US for the canning industry to adopt conservative heat treatment process, which reduce the survival of botulism significantly. So even for the canned food, that how the requirement of the heat, how the food can be protected from the Clostridium infection. So that has been finally discovered in 1920. And after that, there are food processing industry also, you know, uh, having the similar kind of facility, having the pasteurization, having the heat treatment process to preserve the food. Also, they use some preservatives so to extend the shelf life of the food item. So, what is the causative agent? Causative agents are basically not only the bacteria, it could be parasite, virus, even yeast, the fungus, even some other items as well as the molds, right? And these are very tiny and teeny, like it cannot be visualized in the naked eye, even until it forms like just mold kind of mesh-like structure of the what we call sometimes a colony that is currently visible. Otherwise, one tiny single cellular unicellular microbe is not at all visible in our naked eye. And certain type of organism, like for the bacteria or fungus, that is not actually causative agent. Basically, they produce the poisonous toxin that contaminate the food, and that is why you guys fall ill. So contamination can also occur at home. It is not like that whenever you have the outside food or you are eating the food from street food, street side. So it is not like that. Contamination can be occurred at home as well, where the food is incorrectly handled or cooked. So while it is known that raw meat, poultry, and eggs can also harbor diseases, in recent year, even the fresh fruit or the vegetables also be contaminated, and there will be contamination of the fecal item when you consume this kind of food, raw vegetables, salads. Uh, that time also you can fall sick. So the common mistake that causes the food poisoning, that holding the food in a wrong temperature. So most of the cases now summer season is coming, right? You cannot keep the food for a long time outside. That is whenever you can have the food already, even if you cook, even if you have the food over there outside, there is always chance like the food get contaminated or not contaminated from the air. It is everywhere. The microbes are everywhere, so it can have a chance to grow easily, and that has an opportunity to the favorable temperature to grow. So even the food is outside in the summer season is coming, it can also have a chance to grow, and getting the food is becomes stale, and it is hard to consume. And if you consume this food, you may fall sick. And poor personal hygiene. So that is one of the measures, causative resources where if the food can be, getting contaminated, and the food becomes contaminated with some kind of spore or any kind of germs when people consume. The personal hygiene means people use their just like a contaminated surface, touching the contaminated surface, then touching the food. So that means when people, that food worker or food who are making the food, he or she touch in a contaminated surface or the on the cloths, and again they try to cook the food without washing the hand. There is a always chance that it carry the germs from the cloths to the food. Touching contaminated food and then touching other food, that is one of the option to how the food is getting contaminated, and touching portion of their own body, and then touching food. So that every time you will see that people do not wear the any kind of the gloves they use, and sometimes there is a chance they used to use the rubbing with some kind of cloth, the nasty and filthy cloth, and sometimes there is a chance like some people cough or sneeze, so they use the same hand without cleaning the hand properly, again come to for the processing of the food item and serving you the food. That time there is a high chance that you will fall sick, not only for the any kind of the stomach related problem. You may fall sick by the food contamination, even for kind of respiratory disease as well, because respiratory disease mostly it is coming from the contact basis. So where do the microorganisms come from? That is everywhere: the soil, plants, utensils, if the utensils are not properly cleaned, gastrointestinal tract, these are all kind of microbes we hold, food handlers, animal feeds, animal hides, air dust, everywhere the microbes. And you must be surprised to know that how many cells we have. Even we have the more than a trillions of microbes in a human body, right? So even number of microbes we hold, that is much more higher than the number of cells we have. Right. So even that occurs, not only that we have a disease, so we can also have a commensal mechanism and a symbiotic relationship. Without a microbe, we cannot, survive. So, but microbes are very much opportunistic, and also microbes coming from outside, there is a chance like it can have a any kind of immunocompromised people that can easily get infected. So here how the fecal contamination can poultry, dairy cows, human, sewage system, if the food item and the drainage system, it goes to the agricultural field, and also cattle, poultry, if those also the poops can also contaminate the food item. Even for the manure soil, that also can contaminate the vegetables, fruits. So there is a lot of ways that food get contaminated, and the fecal contamination is very common in India. Here I could say, simply tell you the common microbes for the foodborne pathogens. Here you can see the E. coli, even E. coli, what we use in the lab, but E. coli is basically is a pathogenic in the soil. Even you can have a diarrhea from the E. coli. Salmonella is one of the nasty, pathogen that causes not only the typhoid, even if you have the cause like stomach cramp, which is known as the salmonellosis, Staphylococcus aureus, that is quite common in the skin. However, that can also gives rise to any kind of foodborne diseases, Listeria, Campylobacter, Shigella, and a virus. Some of the common viruses, the norovirus, rotavirus, and hepatitis, that is quite common, and some parasites. So even people use the pork, the beef. They are also having chance of getting the parasite infection. Not only that, the kids, infants go to the field without barefoot, and also have a tendency to take the food from the floor, which is also we have a chance like this parasite, that worm can be also gone to their intestine. So those are all the ways someone can get infected. So the bacillus, one of the diarrheal, causing the disease causing bacteria, and also not only diarrhea, some people have a like emetic type. That means somebody may have the vomiting tendency. So Bacillus cereus actually, produce the cerulide, that is the toxin. It is not only the bacteria, even the toxin basically is responsible for any this kind of foodborne disease. Clostridium, it is a powerful toxin to the man, which is a key virulence responsible for the pathogenesis. And another, this kind of toxin is very much heat resistant, the Clostridium. So I will coming to the point, what is the best method in the last, it is a good question, Priyanka. So I will definitely come to the end of my, slide. So what would be how you can avoid the disease, and what would be the best method? So let me finish. The Clostridium difficile also found the feces of the human. So 35% of the human also contain. Okay, so this is the two potent toxin when the food is getting contaminated with this Clostridium, and perfringen, that is also one of the Clostridium species that also causing the several foodborne diseases. So that the foodborne diseases mostly again coming not only from the bacteria. Again, my concern is that toxin which is heat resistant. So there is a chance like the spore can be broken. However, the cleaning of hand, and how to avoid, not only by the cooking the food in a very high temperature, how to get rid of those things that you have to try to avoid. It should not get contaminated through any kind of fecal uh, contamination should not happen. That is also one kind of vital portion. And those are also acid labile. So those pores are also acid labile. So in that kind of cases, that food can be protected not only by the heat treatment. You can have a acid, because some of the acid serve as a preservative. You can see why pickles are stable for long times, right? So pickles are very much acidic. So in that acid, so that there is a high chance this kind of bacteria cannot survive. So that is one of the way that, South Indian people, there is a high chance that, because of the heat, there is a high chance that fermentation generally occurs, and also they can also protect this kind of pickles. They protect any kind of microbial contamination because bacteria favors heat, and around 37 to 40 degree, which also is a favorable temperature for the bacteria to grow. So they generally consume the pickles and some other fermented food that can have a longer shelf life. Okay. So coming to the Campylobacter, which is also one kind of microaerophilic gram-negative and non-spore forming bacteria, which is also requires the temperature, say it is 41 degrees Celsius. So that bacteria can also survive. So freezing portion and refrigeration is also one of the way that I will come to the, after a few slides, that how you can, stabilize that food item for a longer time period. Salmonella is also one kind of zoonotic disease because Salmonella mostly come from the birds and the chicken and mostly the egg. Okay, and there is a myth people consume the food like eggs, that is called raw eggs, to gain some kind of dragon-like energy. But if it is contaminated with the Salmonella species, okay, so these also having symptom like salmonellosis, the stomach cramp, and the bacteremia and diarrhea, and some kind of salmonella also causing the disease called deadly typhoid. This is a fatal disease. Okay, the main contaminated food of the salmonella is the poultry. However, that from the poultry, when the fecal contamination comes from the bird and also comes to the field and the vegetables, even through the spices, the MDA spices is banned in California because there is some kind of salmonella spore they found. Even that salmonella can be contaminated any other spices, even vegetable item as well. So if the Salmonella, when see in the UK, in the few years back, there is a outbreak, Salmonella outbreak that is coming from the green salad. So mostly the green salad is contaminated with any kind of fecal contamination from the birds, or the Salmonella, affected any kind of contaminated surface or the stool, right? And which is called the zoonotic disease. E. coli, as I said, that E. coli 157 H7 is causing the hemorrhagic diarrhea, and it is very much common, the bloody diarrhea in Indian population as well, and it can contaminate the milk, meat, fruit everywhere. Then why people eat it, why you people eat, there is a myth why people eat uh, the what is called contaminated food, that is unknowingly people eat, definitely. That is quite common, good question, that of course people are not sure if somebody food is being provided from to the restaurant, definitely you will consume. You never know that food is contaminated, right? You need a food item and you are having the milk because you need to grow up, and milk is one of the greatest nutritional source. Okay, so the food, the milk has to be consumed, but in the proper temperature, proper, that is also needed. Why we generally boil, even the milk is also coming not like, from the cow itself. It is going through the processing, what is called the pasteurization technique. It is coming as a chilled technique. So I'll come again. Um, that is the technique where the food, this kind of can be liquid food can be disinfected. The meat, remember, while you process the meat and fruits, you are supposed to use the pot water, that is your, of course, the clean water, right? So potable water you have to use, and you have to remember, after getting the food, vegetables from the market, you supposed to wash it properly. And most of the E. coli contaminates that food does not taste and look and smell bad. So even it is the stale food looks very bad, too hard to consume. Definitely, you will be cautious not to consume. But if the contaminated the E. coli, how you know, right? It occurs like unknowingly. Listeria, one of the disease that listeriosis, which also uh, uh, contaminated the vegetables, milk, cheese, and it causing the septicemia and also miscarriage, meningitis, diarrhea. This is very common, symptoms. And as I say, the pasteurization, okay, that food contaminated with the listeria can reduce the count of these bacteria. Okay, so not all the people will fall sick. Again, coming to the point, the same food you both say two-person consume same time, but it is not like that. Everyone will get sick. So it is as per the immunity, how the immunity works, and it will kill the foreign pathogen. But in that case, in your case, maybe you are a little bit of immunically down, so in that case, bacteria have a chance to grow further in the gut, and you will fall sick. Vibrio cholerae, that is quite common disease, and even many people died, and that occurs in the pandemic occurs in India itself. And Robert Koch discovered this, that cholera, that bacteria is a Vibrio cholerae, is the main, causative agent for the cholera, where the Somunath, they one Indian. Uh, am I audible? Hello?
Chiranjit, you are audible.
Yeah, because I got a notification like there is a connection of the, failure from my side. So that
No,
is
no,
why.
we so far there is no interruption at all
No
from our side.
interruption, right?
Yeah,
Okay, okay,
yes, yes.
okay. Thanks for letting me know. Okay. So that the cholera, how actually it happens, we do not have that time that even still, I should say, India and a third world country, that developing country, do not have the water which is coming from the faucet, which is not clean. That is why we have to have our water purifier system uh, in the in the in our home, right? In our every houses. But in a Western country, whatever water even supplied, that is cleaned water. Even whatever water you are actually taking for shower, that is also you can consume. So that is unfortunate for us. And earlier, what happened where the kettles are taking bath, and also people are drinking the same water, and where the river is being used for cleansing the clothes, people are consuming the same water. So people do not have the idea what they are consuming it, and there is a like severe pandemic occur, like many people fall sick for the cholera, like plague also one of the pandemic occurs earlier. Okay, so this kind of disease occurred earlier. Nowadays, it is quite, subside, but still in the village where the poor health and hygiene, and that is your responsibility or the student's responsibility, that can also teach. their relative parents and still in the village uh, should not consume this kind of water, otherwise there is a chance to fall sick. Shigella, one of the, are you saying anything? Okay.
Yes, we can see.
Okay, sure. Shigella is one of the bacteria that also causing the bacterial dysentery. Okay, that is also quite common in US and some kind of, still what we can say, even Western countries also fall sick. It is not like, third world country in Africa and India like. They also consume several food items which is contaminated with many, bacteria, viruses everywhere. And a common food item, even those are sold in the market, like chicken salad, the salad in this even sold in what is called DMart. You can find salad over there. So there is always chance the contamination may happen even if it is not processed properly, not properly handled. Who is cutting the salad, his or her may be contaminated, and the fecal material and unsanitary handling of the food worker, that is one of the major transmission route. So that also can happen. So it is always better you can bring the food and you know what you are doing. You can have a proper surface. You can cut with your knife, which is also properly cleaned. You wash your hand properly before cutting the food. So those are all pathogens, and you can see that this pathogen how it causes per year, that is globally. I mean, this much, 200 to 1000 death. Even for the salmonella and typhoid is very common in India. Even for the salmonellosis, those are having the stomach cramp, Staphylococcus aureus, E. coli. These many deaths occurs every year globally, and not only the bacteria, some virus. So again, that is virus cannot grow, right? Okay, so even virus, if it is in the food, it does not grow. But if you consume the food and with the virus, so virus will find a host. So after that, it will replicate. It will have a chance, like good sources like nutrient, because that now virus find a suitable host and it can grow. Okay, so hepatitis A is not like a hepatitis B. Hepatitis B is not a very common. Of course, it is deadly, but hepatitis A, most of the students, even for us, also suffer, which is commonly known as jaundice. And people suffer the jaundice who are having the hepatitis A infection. That whole liver got actually damaged, and you have to in the whole lifelong, basically you have to be very very cautious about that, for the liver function. It cannot be recovered 100% what was earlier. And the food worker, the major resources of the this kind of hepatitis A virus. And the common food, as I say, the salad. Of course, people here in India's context, the people consume the golgappa, pani puri outside, so they also have a chance like hepatitis A contamination. If you survive, you are lucky. If it is already contaminated, because you know the hand is not properly cleaned, and you might have seen that the same kind of water, the tamarind water people use, and that can be like for years they are the seller basically using it up. They do not throw it away until unless it is gone very bad. So there is always chance you can have a stomach upset or any kind of diarrheal symptom. Okay, and if you have a like hepatitis A, so then that is very dangerous. You know, the jaundice, everybody know that you'll have like yellowish stool, your hand, everything will be, you know, that pale yellow. You'll have, and you'll be very, very tired. You'll have a fever, and you have a large, you know, stomach because of the, you know, the liver, enlarged liver. There is a transmission occurs by the fecal contamination, cross contamination also happen. That not like, you know, fecal contamination, contaminated thing you are directly consuming. It can also having like cross contamination. No, norovirus is very common even in India as well, and also involving up to 3,000 people can be affected. That is by the very much, you know, stomach cramp. This thing happen when you consume any food. After that, you have to simply, you know, sit. Then sometimes you can have a like some kind of antibiotic you try to consume, but it sometimes it will not help because of the virus, but sometimes it helps if you consume some kind of probiotics to subside, because virus things cannot be, you know, eradicated by any antibiotic. It will only go away by taking lot of drinking lot of water, and also the ORS, that, you know, electoral water, if you consume, and after that it will eventually subside after 3 days. But again, how you can prevent: the handle and cook food properly, avoid consumption of the raw seafood. Okay, and the people use that, you know, the prawns, shrimp, use the seafood, and sometimes there is a chance it is not properly cooked. And of course, good personal hygiene, that is one of the major way how you can get rid of foodborne diseases. Rotavirus is very common to the infants. Even India also developed the Rotavac, the vaccine, indigenous Rotavac. India also developed, but it is very common in the infant, because infants generally, you know, touch and, you know, uh, just simply lick any kind of surface, right? So you have to be very careful if in your family the any kind of infants are there. You can go next time and see if any infants are not taking any kind of food item, or any item, not maybe not be the food item, from the floor. And also he should not, he or she should not lick any of the surface or any kind of toy. So that time, rotavirus germs are mostly found in the person's stool, right? But you never know, this can go from the bathroom to the diaper changes, and also it spreads through the floor, and is also contaminate the toy and other material, and where the infant can get infection. Then next comes is the food spoilage. What we have now discussed, that is called the foodborne disease. However, the food spoilage is very common when you keep the food in the wrong temperature, or outside, or not properly maintained. Definitely, you would not like to, you know, consume, but that will not be good, because that will spoil many kind of food items. And after that, that is a global kind of economic breakdown, right? Even for the that food processing industry, and how the food are also coming bread. Even you can say that strawberry, how you can store the, you know, the strawberry for longer time, still in the research going on. But definitely, by the if the food will get spoiled so soon, definitely you'll have the economical problem. So the food has to be in having the longer shelf life. Okay, so the mold is one of the, you know, major you know what is called resources, where you can see that food item becomes the colors, the white color. After that, you'll see that is not at all edible. You have nothing except to throw it away. That is called mold, and it can grow even the wide range of pH 2 to above pH 9. Even the strawberry, you see this how it looks so bad. Still, it is actually showery. Still, the molds can have to grow easily, and their temperature range also even lower. Even if you can refrigerate this item for a long time, even refrigeration for a long time, it is also bad. Everybody have a fridge in our home, right? So even if you refrigerate your food, you are not supposed to consume let the food for a long time, like for 7 days or a month. Even if you refrigerate this kind of mold, and also food can also get spoiled easily. Molds can also grow in a limited moisture, so that is one of the drawbacks. Even some of the less moisture containing food item can also in having the mold. So mold, some of the molds, even if you do not know, some people, most of the case, your mother do not like to, like to throw them away, do not like to waste the food item. Sometimes what happen, they generally cut, and they generally not let their kids to consume, rather they also try to save the money, definitely not to waste the food. Definitely it is a common practice for the mediocre, any kind of mediocre family, that how we can see. But some of the molds are having the toxin, and this is one of the toxin that is called aflatoxin, that causes the aflatoxicosis, that people may get hospitalized immediately after having this consumed by the aflatoxin, right? And that also you will find this aflatoxin is very common, not only the this strawberry or the aspergillus, this kind of fungus mold species. You can have, like the corns, other pulses you can find. Another thing, it is aflatoxin is very common, that is called red chili. Okay, the red chili people generally go for in a village. You might have seen the red chili, and they generally keep it outside for drying purpose. And some of the chili, some of the peppers get having the fungal mold contaminated, and those are having the good sources for the aflatoxin production. So beware of these. The red chili peppers are one of the major source for the aflatoxin and infection, and causing the disease, uh, aflatoxicosis disease. Okay, the prevention of aflatoxicosis, the proper drying and storage of grain and other affected food, even as I said, that if there is a chance any kind of molds are there, it is better to just, throw them away and segregate which is not rotten and not infected with the molds. Otherwise, it will spread very fast. And quality control of potentially hazardous food by use of appropriate analytical test. So that also, definitely it is not possible for you at home, but those are in the the food processing industry, they generally test. And also some people use the fungicide as a preservative while seed dressing, and like pulses, potatoes, against fungal invasion. So that is quite common practice while before packaging of the food material. Next coming is the foodborne parasite. So not only the bacteria and fungus, as well as viruses, parasites are also very common, that causing the foodborne diseases. Here, the giardia is very common in India's context, that people do not, wear the shoes or the chappals. Mostly the parasite, the giardia, and entamoeba, this kind of worm coming from your bare feet, and it can directly infest inside. Also, the raw food, vegetables, even people still 60 to 70% people still, go to uh, the field for defecation, still in the village. Some you might have seen some kind of movies, I guess, that still the defecation of still there, and they do not go to the proper toilet. So in there is a chance like the fruit and vegetables are contaminated with the giardia, and it also contaminate the water system. And when some people drink this water, you knowingly or unknowingly, definitely unknowingly, because it will definitely, after rain, it will contaminate the rest of the field water system. And when you even bring those food item, you never know that it is already infected. Toxoplasma gondii, those are having the pest. Very, very beware of this. Cautious. People generally, kiss even the cats. I mean, there is a chance like cat feces contamination also comes from the fecal to oral contamination generally happen by this route. Toxoplasma. How to get people toxoplasmosis? Toxoplasmosis is actually disease, not only by the this cuddling the pet item, by consuming the food, and again the raw food, pork, lamb, and raw uncooked meat. Also, this very common. And how you can definitely prevent, if you can see that temperature around 145 degrees Fahrenheit, right? So minimum this kind of temperature is needed. You might have thought like, definitely when we cook the food outside, sorry, in your home, definitely the temperature will be more than 63. However, when you take the food item outside, there is a chance like um, is not cooked is properly. And even some food is cooked properly, and you are taking the raw salad, so it does not matter. Even suppose you are taking some kind of food item which is very hot, right? Some kind of like vada pav. Now you take with some kind of salad which is contaminated. It does not matter. You will be affected whether you are taking that very hot vada pav or not. Okay, and you definitely washing of the hand thoroughly with the water and soap. Okay, even we use the sanitizer, but there is no alternative of the using of the soap and water. Even some people simply cuddle cat and the dogs and the pet animals, and after that you have to wash your hand properly, and also after touching any food item. Then Trichinella Perilasis, that is one kind of worm disease that also occurs that undercooked meat. That is also the same kind of transmission. Also, it is very common. Then teniasis, that tinea solium, one of the major cause that of the disease, causative agent of the teniasis, tinea solium, that is very much common who usually eat pork and beef. And how you can definitely prevent the teniasis, not only using the roast in a minimum temperature that is mentioned over there. However, as I said, the cattle, the poops are around, right, everywhere. So even also dogs, street dogs, also having the poop in the roadside, in the same chappal, same uh, uh, shoes, you are actually coming with uh, contaminated fecal item, and you directly go to the bed and home, and this worm also you are actually carrying with you, right? So there is always chance. Excuse me. Hello. Yeah, just for 5 to 5 minutes. Okay, sure. So, am I audible or hello? Am I audible?
Yes.
So this kind of spoilage also occurs, but however, you think like, okay, you may have the food item which is contaminated with only very few amount of bacteria or some kind of, disease causing agent. However, you can see within few hours the bacterial population how it grows, right? And when there is a chance like it will a particular temperature it will reach. So here it is called that within that few minutes, the several bacteria will be populated. Okay. Then what we generally call that what lays those organism for the favorable temperature, not only the temperature, water, moisture, time that requires those organisms to grow, right? One is the food which supplies the nutrient, then acidity. As I have shown you, that is in the even for the broad range of acidity, fungus can grow, but not the bacteria. Temperature is the one of the favorite temperature. If you cool it down, it will inhibit, but it will not let the bacteria to lie. So it will have a less number of bacteria. Then time. Then suppose you wanted to have the fresh food after just after cook. So then you have a chance like less infection. Definitely, if you keep the food for longer time, you are letting the organism to grow. Then oxygen is for us and for the all kind of organism. Oxygen is one of the major source that how the bacteria also survive. Then it came like canned food you will see that canned food can be stored for a long time. Then is the moisture actually favors the any kind of growth. Have you ever seen the biscuit or any kind of dry food that is contaminated? It is no, right? Because it is a very low moisture is there, so that does not let the bacteria or any kind of organism to grow. So that is that what I said, the food is the nutrient, acidity, different bacteria and fungus having they can grow in the different pH. So if the food processing industry can maintain the pH, then they can get rid of this kind of contaminants. Temperature, that is why we are using. हाँ तो क्या करें अभी because I have started late, right? No, this is Chiranjit here. Yeah. Yeah, can you please? Sorry for the interruption. So, am I audible? Can you please turn on my microphone? So again, that temperature is one of the major causative what is called physical parameter, as you can say, that how the microbes can grow. Then time, as I said, oxygen and moisture. As I say that those items, those are having very low moisture, and that cannot have it let the bacteria and other organism to grow, and that can stay longer. Of course, you can consume the biscuit even after 1 month because having the low moisture. So you can have a like that what is called water activity. So even zero point 0.3 water activity, no microbe can proliferate. And you have to remember that term is called fat term, that food, acidity, temperature, time, oxygen, and the moisture. So that regulate, and that how the food processing industry also, consume, take this kind of uh, mm, take this kind of physical parameter for to have increase the shelf life. Right. So I should go fast. Okay, so then again, the last is the prevention of the food spoilage, the cook food thoroughly, practice good personal hygiene that you have to maintain whenever you take the food, even in your school, colleges, even offices. Good personal hygiene always needed. Clean, sanitize food contact surface after use with the raw food. Even if you have a raw food you are bringing from the market, so while cutting, and you have to wash properly. Avoid use a raw milk product. Longer storage must be avoided. As I said, even if you refrigerate it, put it in the fridge, still there is a chance like the food can, microbial contamination can be taken place. And store the food in cool and dry place. Avoid cross contamination. That means not like contaminated surface you have to, use. Avoid consuming cut fruit. Generally, you have the nira outside, and nimu pani outside, and this is the summer season. Of course, we all consume. However, there is a high chance like people will have a diarrhea. Cleans arms properly after touching with cats and other domestic animals. That is also one of the vital things that those are having the pets uh, at their home. Okay, thank you so much for paying attention, and I hope that you like.
Awesome, Chiranjit. It was awesome. Very nice presentation. I have learned myself a lot about food and other things. Really, I used to, depend on cut vegetables a lot. So I think I have to be very, very careful. And thank you for such a wonderful talk. And I'm sure most of the students, some of them whom I had met when I had gone out of town where there is no lockdown anymore, they told me that they even started cooking in the kitchen. Their parents used to tell them, come on, you're at home now, try out something. And some of them have also experimented, some of the experiments they have done. So I'm sure they all are very much aware about this talk and they will never forget about it. In case students, your friends have missed out for some reason or the other, we do have a recap of this on our CSR Jigyasa YouTube channel. Please go and watch that. And thank you very much, Dr. Chiranjit, for taking your valuable time out and coming and talking to the Jigyasa students. We would like to have you, connect with us for many more such different topics on
Absolutely.
biochemical sciences, biochemistry. It's a favorite topic of everybody's. So thank you so much.
All
So
right.
with this, I would like to now just quickly show some of the experiments by my team. And I know that most of the students are going to tell me, what ma'am, you're showing this, we know it, we have done it. But something, students, my team, they are MSc and all, and they don't know. I want them to learn. So I said, come on, you all show these simple experiments to my students, and then you see who is smarter, the students or you. So before we begin that, I would just like to take some of the questions if they have put up here on our chat box. And, I would like to request Chiranjit to please answer those questions. I think on YouTube, I looked for it a while back and I couldn't find any. And
Should I?
students, you can also raise your hand, student, and we will unmute you and you can yourself ask questions. All right. So I think we will just go to the first question.
Should I unshare? Uh,
Yeah, you can unshare.
Share basically.
Yeah.
I don't know. Okay, stop sharing. Here you go.
Yeah.
Okay.
So I will just check if anybody has sent any questions here. Hmm. Sir, what according to you is the best method to avoid microbes as foe so that they can act as friend? Priyal Khanna wants to find out this.
So she actually asked me at the beginning, right?
Okay.
I don't know whether she have gone through my all my slide, or
Yeah, maybe it is answered for her, but still, Priyal, if you want, you can, raise your hand and we will unmute you. Do you still have the same question with you? So I don't see your hand up, Priyal.
Which type of bacteria used to make cheese and butter? That is called the lactobacillus, lactic acid bacteria. Mostly people use.
Priyal, you are unmuted. If you want to say something, you can.
Ma'am, now my doubts are clear.
Okay, very good, very good. Did you like the talk?
Yes, ma'am, really.
Okay, okay. Anybody else wants to ask, please let us know and we will unmute you. We have got Chiranjit, we have a very young student with us. And since we do not have any questions there, just let me check YouTube channel if there are any questions. And then I'll tell you about this, what I was telling you about this student. So Rohit, are there any questions there?
Yes, ma'am. One more is there from Priyal. So can I dictate it?
Yeah, yeah, you can just
Okay, so I'm asking Priyal Khanna's questions only. So
yeah.
what according to you has less amount of microbes and best for human body, whether it is a
Oh.
green vegetables or non-veg, and why? So
No, it is not like that. That the microbes should present in the meat and less amount in the you know green vegetables. So there is a contamination occurs from everywhere, right? And that is like even I'm just talking about say salmonella. That is a chickens and that mostly the reservoir. Okay, but salmonella is also infect chicken as well as that some zoonotic salmonella also infect the human. But if the poops, right, it is contaminating with the eggs, and also the same poops are coming to the vegetables, it does not mean anything like, vegetables are not having the less amount of salmonella. Even it can stay for longer time for particular temperature, favorable temperature. It may not grow, may not replicate, but it will remain as it is. And when we consume, after without washing and not properly washing or raw salad we are consuming, as I said, like salmonella contamination also happen in the MDH spices. So that has been globally now in the US market. They sometimes banned Indian spices one time in California.
Mm.
So when we are there, because salmonella outbreak is very much common in US, but some kind of salmonella species also here. Mostly the typhoid is very common over here. So it is not like the bacteria cannot, even for the desiccation, still it can survive. So when we consume, so you will fall sick because now is a favorable host, like human is one of the host, right? Now it can have a chance to replicate and it can grow easily and even the desiccation, it may still just like a forming the spore, it will remain for years. After that favorable situation happen, it may start replicating it and you will fall sick. Of course, the number of bacteria, if you definitely, our immune system definitely try to fight with the foreign body, right? If you have a less bacterial load, less virus load, definitely you can survive, right? You may not have any kind of symptom. It does not mean that you may not have the infection, right? But you may not have the symptom, and our immune system can fight and get rid of and lies the bacterial system, lies those that viruses. But whichever there is always chance like fighting occurs between the prey and predator. So who will win? Because more amount of bacteria, if you stay the food, if you keep the
क्या
food
बोले?
for longer time,
Sorry.
still there is a chance like number of bacteria also being increased, right? Favorable situation. So even if you consume, then you have a chance to fall sick. I hope that answer you.
Yes, thank you so much, Chiranjit. And we have got one more question. One person is asking
Hello.
that there is bacteria in fast food, and then why do people eat it? Well, we all eat, I think, fast food,
We all
pani
eat.
puris and other things.
We
Yeah,
all eat. Oh, but this is
we all
kind
eat.
of factor. Like people are going for the Nimu Pani, Nira is there. Even even for me, I got sick for few in the last week. The severe, food poison happened. What I suspect that coming from, I went to the one temple in Mahalunge. Then I, drink those sugarcane juice. So there is
It's very
always
hot and summer.
it's
Yeah,
a
it's
hot
very hot in
summer.
Pune.
Definitely, it is good. But of course, if you bring the pani puri over there from your DMart or in the store, and if you can make it at your home, so definitely you will be more cautious and you will have a less, chance of getting infected. If you
Definitely,
can make
definitely.
it at home in a condition that is a hygiene condition.
I think this question was taken, Priyal. I'm sure you say that according to you, which has less amount of microbes and is best for the human body. Green vegetables, non-veg, chicken, mutton.
No,
Why?
that is
And please
that I
explain with
already
respect to
said that it is
microbes.
not like the case. It could be anything, like even the agricultural field also contaminated with the cholera one time, because people generally, even for today also, in the village or the very remote village, who prefer for the do not have the toilet till unfortunately go to the field. So it is not like that somebody consume the direct feces, but getting the vegetables and consume the contaminated fecal contaminated vegetables.
So we have one question from Akshay Kamble. He says, "Sir, which can you tell which type of bacteria is used to make
Yeah,
cheese
I
and butter?"
already
I think
answered. I
we have
already
got a
answered.
quiz here, but he's quizzing you, Chiranjit. So have you addressed this? Which type of bacteria
Yeah,
is
I
used to make
already
cheese
told
and
them that it's called
okay?
lactobacillus. Mostly people use for the probiotic purpose. Okay, but there are also true like lactobacillus are beneficiary, but this is called, we all as a microbiologist and even for you should know that everything is opportunistic. So even we have the commensal bacteria, those are we generally use for ferment any kind of dairy product. However, certain pathogen that can also be pathogenic. So you have to remember, right? Even people use for the cheese batter for the lactobacillus species. So that is quite common bacteria. People use the cheese and butter.
Sure. Thank you so much. So Rohit, are the questions from the YouTube channel answered? Have you taken up
Yes.
the questions, Rohit? Okay,
Yes.
sure.
Yes. Yes.
So now, without taking too much of time, I just want to introduce you, Chiranjit, to one student. We have one international student with us today.
Oh, really? Really?
Yeah, and
Great.
her name is Zara, and she is
Okay.
here. So I just want to find out, Zara, would you like to
International.
say something? I will unmute you and welcome,
Hi,
Zara. And hi, how are you?
good. How are you?
Yeah,
Where
I'm
are you
good. Thank
from?
you, dear.
Thank you so much for this opportunity.
Where are you from?
Australia.
Australia. Oh, great.
hmm
Australia, where?
Uh, Melbourne.
She's from Melbourne. And Zara, was this useful to you, dear?
Yes, very useful. Actually, it's gonna help me in my learning.
Really? So,
Great.
how about any food poisoning disease in Melbourne? Do you suffer any kind of food poisoning disease over there? Or your parents?
Mm, we I don't know.
Don't know. Really? You never had any kind of stomach cramp or vomiting, any kind of diarrheal symptom over there? Really?
No, I don't really have much of like. I've not really come across of any of these.
Really? That is very great then.
Yeah, very lucky.
Very lucky. Might be your friend might have suffered.
Maybe, maybe.
Sure. Thank you so much, Zara. I'm sure your mom is a very good cook, and that's why, you have been taken very good care of. So thanks a lot for joining us. And thank you, Chiranjit. We will now go to our students. I told you, kids, that my students are just learning from you all. Okay. So please guide them and please help them. So we have got two students, Abhishek, and one is the other one is, Manisha. So Manisha and Abhishek, are you all ready? Let's finish this quickly so that we can go on for the quiz. And I must first and foremost thank Chiranjit for setting up the quiz for us and giving us the questions so that we could arrange today's quiz competition for the students. So immediately after the science experiments, 2-3 experiments which you all already know, please put up your hand and let us know how you can make it better. Let us know if Manisha has done any mistake. I have told her, Manisha, please do it properly, and I'm sure she will do some mistake, and Abhishek is here to kind of just add to that conversation while the experiment is going on. So over to you, Manisha and Abhishek. Please put your camera on and say hi, and then you can begin your experiment. So quickly, we need to finish in 10 minutes.
Okay, ma'am. Am I audible, ma'am?
Yeah, you're audible, but we couldn't see you. Yeah, we can see Manisha, this container, and now we can see you. No, more up. Yes, you look good today. Yeah. So this is Manisha, and she is already done her MSc and she has done her internship, and she is with us. In our Jigyasa team, she has joined as an outreach, what can I say, program coordinator. And we have Abhishek, he's our admin coordinator, and he's going to help and amuse us. And let us see what they both do as a team. Okay, so please hurry up. Let's do it fast.
Hello, students. Good afternoon. So today we are planning some a few experiments which you can try at home to explain some basic chemistry. So as an introduction, I would like to remind you of the concept of acids and bases. In lower classes, we have studied that acid is something that tastes sour, and so base is something that tastes sour and the soapy in texture. That is the basic things we study. So I'm keeping it like that. And now we also know about indicators, which will help us differentiate between acid and bases. So two of our experiments are based on natural indicators. So natural indicator is something that we can directly take it from nature, which help us to differentiate between acids and bases. Today we are using two natural indicators. One is turmeric and another is beetroot juice. So I think we can start the experiment. So our first experiment is using turmeric, and turmeric, I think everyone is having at home. So can you see this?
Yes.
Okay, so students, can you tell me what color do you see?
Oh yeah, I think they won't be able to talk, but they can chat it out. Students, you all can chat it out. What color do you see? Yes, Manisha, go ahead.
Okay, so I think you're seeing a yellow color. So this is the color of turmeric, as you all know. Now we are adding some water to that and making a solution out of it. Now we have a yellow solution. Okay, so this is the natural color of turmeric. Now we are adding some soap. Okay. So can you see this?
Oh,
What have happened?
this is like homemade magic.
Yeah. So
Looks like a dark wine color,
Yeah.
yeah.
It's
Now,
dark red.
yes. So what have happened is turmeric actually turns to red color when it is in basic conditions. That is why we are getting this red color. So now we can understand that soap is a base, and this because of this turmeric we can understand that it was base. Okay.
Okay.
Now, what if we add some acid into this? Do you know what will happen?
And what is the acid you are using here?
I'm using some lemon juice. I'm adding some lemon juice.
Okay.
What have happened? The yellow color have came back.
New. Yeah.
Okay.
It seems like,
Abhishek, tell what you are observing.
yeah, I'm seeing it the before color.
Okay.
It turns yellow again. It's
Yeah.
like magical moments.
Yeah. So what have
It
happened
is
is
red and again it seems to be a yellow color.
yellow. Now, when we added like acid, what happened is the base got neutralized. Now the situation became acidic, and turmeric will give yellow color in acidic and neutral conditions, and red only in basic condition. That's why the yellow color came back.
Yes.
So that was our reaction with turmeric.
This is the first one, no?
Now, first when we added turmeric with detergent powder, we got red solution. Then we added lemon juice to it. Then we got the yellow color back.
Red solution turns into yellow.
Mhm.
We put the acid in. Okay. Student got that?
I hope everyone
Yeah,
understood
you
it.
can easily access these experiments at your home as well.
For the next experiment, I would also like to revise you about the topic of pH. So pH is a measure of acidity or basicity. In the previous experiment, we just differentiated if it is an acid or a base. So what if we have different acids and bases? Then we can characterize if it is less acidic or less basic. We can characterize it like that. So pH is a measure of acidity. Like in this chart, you can see this is the whole range of pH from 0 to 14. 7 corresponds to neutral, and less than 7 is acidic, and more than 7 is alkaline or basic. So as you can see, the stomach acid. Do you know what acid is present in stomach? This
No.
HCl.
Can you please see which acid is there?
HCl is the acid present in stomach.
Okay.
So that is very acidic. And here lemon. We used this acid just now. This citric acid. This is also a very acidic, a very strong acid. Then moving here, we just use soap. Soap is very basic. Okay. And the neutral water is and the pH is 7 and it is neutral.
Okay, that's why water has no taste, okay? Because it is neutral. Okay.
Okay. So these are the color when using a neutral pH paper. Now we are using beetroot juice as the indicator to show you some color variations. For that, we are making some 4 different experiments. Hope that everything is visible to you. Now, can you see this solution?
This is
Yes,
what?
we can see.
Okay. Okay, what is the color? Can you see the color?
Yes.
Okay, what is the color?
It's light red or
Yeah, light red.
Yeah.
Do you know what the solution is?
This is the solution of beetroot.
Yes.
How can it be extracted? Can you please explain?
Okay, for this, I have boiled some beetroot chunks in water for like 10-15 minutes,
Okay.
and I have extracted that liquid part, leaving away all the beetroot chunks.
Okay.
So this is
So students can get this easily accessible to on their homes also.
because beetroot is in everyone's home. Now we can prepare this juice also easily.
But students can also make it at home.
Now what I have done is I have poured beetroot juice to 4 different bowls. Okay. And
What are you gonna serve?
I'm going to show how this color will change on different acids and bases.
Okay.
Okay. So for first one, we are pouring some.
This is like you are going to check the reaction of base and acid to the neutral indicator, that is we are having the beetroot juice. Okay.
Yes.
Beetroot juice हमें easily accessible है, आपको ये easily मिल भी जाता है. So you can access it on your homes.
So in one of the bowls, I am adding some soap, and can you see the color change?
Yes, it's like dark wine or dark red.
Yeah, turn to a darker color.
So हमने इसमें अभी सर्फ पाउडर और कोई भी सोप पाउडर का एक्सेस किया है, तो आप देख सकते हो it has the deep color while we are having the reddish one, which is neutral or normal, and this one is after basic solution. Okay. After that, what are we adding?
Some baking soda powder. Baking soda.
This is baking soda.
Yeah, this is baking soda. I'm adding baking soda to another bowl. Can you see the color change?
It's like a deep red color.
Yeah.
Students, you can also join it on chat. Do you see the color changes?
So the earlier one, I think, is more dark brownish or dark deep red, and this one is a little bit maroonish, the second one.
Yes. So there is a difference, no? That's why this is more strong base. This is a weak base. That's why the color difference is there.
Okay, then surf powder is a strong base.
Yes. Now, in another bowl, I'm adding some lemon juice. Let's see this. I hope you can understand the difference.
Okay.
What color can you observe?
This is little dark red. I know.
This one?
Yeah, it appears to be a little bit dark red. The other one is a bit maroon, and the first one was a dark, very dark brownish maroon.
Yeah, there is a gradient now, no? Dark, less dark, again less dark, less dark. Like that.
Right, right.
Okay, for
so this is an acid.
So everyone to the different colors.
So what happened, Manisha? Why are these change in colors? Can you quickly tell us?
Yes, ma'am. So here we are seeing a scale, like this is in the presence of acid. This both are bases, but their basicity is different. This is a less weak. This is a weak base, while this is a strong base. That's why there is a difference even in bases also.
Correct. Yes, that's right.
So
okay. So now this
so are you done?
Experiment is
I have one more experiment.
And the last one.
Very good.
The last experiment, we are preparing this vinegar, baking soda, volcano reaction. I'm sure that many students have tried this at home or school exhibition or something. For that, we are taking a small bottle and adding vinegar into this.
Okay, we're gonna add vinegar.
Yes. This white colorless liquid is vinegar. I hope everyone has this at home also.
Mhm.
Now add some dishwash solution.
I hope it is not dangerous.
No.
Is it?
Most students have done this at exhibition or something for showing volcano.
Okay. Now the second one. What have you added?
Now first I have added acetic acid. Tell me acetic acid is?
Vinegar.
Yeah, vinegar.
Yeah,
Vinegar
okay.
is also called as acetic acid.
Mhm.
So acetic acid is acidic or basic?
It is acidic
Yeah, it is
in
acidic.
nature.
And then I added some soap solution.
Okay.
Now I'm adding some baking soda. What is the nature of baking soda? Is it acidic or basic?
It is basic, I think.
Yes, basic. Now I'm adding
I'm good at it.
baking soda, baking soda powder, and you can see the reaction.
Oh, the volcanic eruption. Student, can you see it?
Wonderful.
Yeah.
Okay, it is erupting like very much. Next, though, it is not dangerous.
It's not
Yeah.
dangerous.
We can use it.
The reaction you see here is the base neutralization of vinegar and baking soda. So then you might think why I added that Vim solution. That is because Vim will stabilize this form. That's why.
Okay, the
Yeah.
soap solution will stabilize the leather.
Lather. If you
Okay.
didn't add Vim, then this lather will not be this stable. It might have disappeared by now.
Okay. So when
So what did you do, Manisha? First, what did you put in this?
I have put acetic acid first.
That is vinegar. Okay.
Yeah, vinegar.
How much ml you should put in that?
Like 10 ml or 15 ml.
Okay, okay, okay. Very good, very good. So students, these are very easy experiments and you all can please go to YouTube and, just watch these videos we have not made, but they are already available. And, we are just coming up with something to make our episode interesting for you. And it will be very nice if you all go and try it out. And, you can also send us your feedbacks after trying it out, how you felt and what makes you, do better and better and how it motivates you to try out newer and newer experiments. So please go to the kitchen, find out all what is available and try to do something. Of course, don't mess it up too much. Otherwise your mom will come and beat me up. Okay, so please do those kits and please try it out. And Abhishek, thanks a lot. And Manisha, yes, you know chemistry. Yeah, I thought you don't know anything. Oh my God, students, they really know. So please stay tuned to us and we will show you even better and better experiments. And you also can try it out. We just want to, try out something from the kitchen because kitchen, it's always full of ingredients unless, your mom decides not to go to grocery and not to bring anything. All right. And she will never do that because she wants you to be healthy and remain healthy. The way Zara, she touch wood, she never had food poisoning. And I'm sure all of you all will love to go and see what all you can do from your kitchen made items. All right. So now I think, Manisha, are you giving us permission to go for our next move on the quiz?
Yes, ma'am.
Okay, thank you so much. So Manisha, thanks a lot. And now we will just go back and we will, start doing some quiz work. So students, be prepared. We are going to do some quizzing and it will be a fun quiz only of 10 questions. The fastest student who will attempt the question, will get more marks. And students who are slower, and even though it is right, they will get less marks. All right, so you have to just start doing that. So let me share my screen. Give me just few minutes and I'll be sharing my screen for, for that, for the quiz. All right. So I think Rohit, you can give them the link for the quiz. And here I will go and put this on. And please give them the passcode also. What you have to do, students, you have to just type on your browser menti.com, www.menti.com, and use the passcode. Rohit is going to put it up. And the passcode, I will call out here 26810015. And just a minute, huh? It's already starting, and I'll share my screen. Not here. Yeah. Can you see my screen, students? Rohit and others, please let me know because I can't hear. There's a loud music here.
Yeah, it is visible.
So maybe some of the students have gone for having lunch because we just crossed the lunch time here in India. It's 1:21 here. So once this screen comes, I can't go to any other screen. So Rohit, you have to manage both the, WebEx as well as the YouTube.
Yeah, okay, ma'am.
So we'll wait for some more students to join. Can you hear the music?
No, ma'am, it's not audible.
Oh, how come this music is not working? I don't know. I have put this music, but I can't even go back now. How many students are there right now on YouTube channel?
11.
Okay, okay. I can see 15 who have joined. We'll
Okay.
wait for some more time and then we'll see.
Sure, sure.
Okay, students, so I'll go to the question now and we'll begin the quiz. I think we have waited enough. We want your full name because if you win the quiz, then we have to give you certificate. So please mention your full name as you would like to have it on your certificate. It says no right answer, but yeah, your answer is right. Okay. And please also send us your email ID because without that, we will not be able to email you your certificate. Okay, we go to the next question now. Please answer this. You get 15 seconds. And if you answer fast, then yes, you will be able to win more points. Okay. Sure, I'm going to the next question, students. Come on, recollect what Dr. Chiranjit Chaudhary has told you. Go to the next one now. Antibiotics are produced by cholera occurs by hurry up, students. Next question. Milk spoilage can be inhibited by. I hope you know inhibited means what? Reduces or slows down. Okay, next question. Salmonellosis occurs due to fresh in your mind, huh? Okay, next question. Hepatitis is what disease? Dash. Choose. Hurry up. Just a small click. Okay. Next, refrigeration. So it's covered towards the end. Tenesis occurs due to consumption of what? Done? Okay, next. Okay, fine. That was the last question. I hope you had fun. Did you have fun quizzing? Immediately everybody said this. So thank you so much for being so spontaneous in this. Thank you very much, students. You all have come here and you all were with us. Really very, very grateful, and thank you for making it a very successful event. Okay, so I will now. Don't forget to join us on Twitter, Facebook, LinkedIn, and Insta. I know that you know what it is. CSI Jigyasa is the handle. CSI Jigyasa is the Facebook, handle also, and on LinkedIn and Instagram also we have the same thing. So with this, I will stop sharing this. So students, this was all for you today, and I hope that you will also join us again in future events. Please follow up us, and even my own handle is @Wafia So I generally, either like or tweet, and if you know that this handle that I am sharing with you, the CSI Jigyasa handle, this covers all the laboratories under CSIR. You know that we are 37 laboratories under the CSIR, that is the Council of Scientific and Industrial Research, and National Chemical Laboratory, that is NCL, where I'm working, is one of the laboratories. And if you, follow CSR Jigyasa, you will be able to go to all the programs that are being organized by all the different laboratories, and each laboratory in India on CSIR, under the umbrella of CSIR, are going to announce all their programs for you students. So Jigyasa is a student scientist connect program for school kids from 6th standard up to 12th standard. So please stay with us and stay tuned with us and have fun. And somebody is asking, when will we get the certificate and the result? So we will email you the certificates. Please give us at least 2-3 days time, because we have to send a lot of certificates, but we will do that as soon as possible. And you will receive this from this email ID, that is oss@ncl.res.in. And if you have any feedback, do let us know and do email us on this. And I'm sure you also know that we have got our own Jigyasa virtual lab. So please go on our Jigyasa virtual lab and follow us, and give us your comments and try out the experiments. Okay. So thank you very much, and thank you to Dr. Chiranjit, who has come here and spent his time, and he was the one who has given us the questions to design, and we have put it up on the mentimeter.com quiz, which organizes quiz, and where we can have this quiz competitions for you. So thank you very much, and be safe and be happy.
Thank you, Wafia.
And
thank you.
Bye all of
Jigyasa
you.
team.
Thank you. Thank you, Chiranjit.
Thank you.
We look forward to you in future. Okay. And thank you, little Zara, for staying with us, and hope to see you in many more programs. And thank you to each one of the students, teachers, parents. Okay. And do come often, and let's have fun the way we had today. And let's teach some science to Manisha. And I'm sure she did a good job. Manisha, thank you so much. Abhishek, thanks a lot. And Rohit is there with us. And Rohit, thank you. He's our IT person. I think 3 of you can come on the, open your camera, and then we will say bye. Okay. All of us can come actually, on the camera.
Okay.
Probably I am not sure about our students. Let me check if I can do something about you here on this platform. If it is possible to show you all here. I can't see you all, or else I could have done that. So Ravi, are you still with us? We have Ravi from WebEx, who was with us since morning. And how do we just show these students? How can we see them?
The student's picture?
Yeah, can we see them?
No, ma'am. You can't
We
see
can't
them
see
because
them.
they
Oh,
have
that's really
joined
too
us
bad.
as a guest.
Oh, that's too sad. Okay, fine, sure. Thank you, students, for being with us, and thank you very much. And I'm sure you all will all be listed. And please see our YouTube channel, and you can see the same event which happened today on our CSR Jigyasa YouTube channel. Please go there and share it with your friends. Okay. So I will just spin up my students here. They will be there. I think Chiranjit is here or no? Chiranjit, are you here? If you're here, then please spin up. We will take one picture. Yeah. Yes. So, Ravi, can you just come? Can you show yourself?
Just a minute.
No, Ravi, you are not still on the video. Zara, can you come? Can you put your video on? If you are still there. Can't see you. Ravi, we can't see you either. So maybe. Yeah, hi, we can see you here.
Yeah, hi.
Yeah, Ravi, we can see you, and I think we can see Zara also. Good, great. I wish kids we could see you. Next time we'll do something, Ravi, please help us to find out something how we can, put the camera on for the students. It is not possible right now, is it?
Yeah, right now it's because in the new update it's not possible, but I will talk to Cisco Tech and then update you regarding it.
Sure. Please don't put so many updates, that we get confused and really we have a tough time with this system. Okay. So thank you very much and have a good time. Okay. And thank you, Ravi. Thank you, Chiranjit. Thanks, students. So let's say bye.
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 Bye.